Bismillahirrahmanirrahim. Another video on the difficult MCQs. This is of course from the November 21 paper 1-2. Please refrain from writing adverse comments, and any positive contribution is most welcome. I'm very happy when you contribute and you say, well, this could be the possible answer. And but I mean, this YouTube channel is to help the A-level students. If it doesn't help one of you or two of you or three of you, please refrain from watching these videos. You can change to any other source which you might find, but I would request you to be a little more have some etiquette and be careful when you write your comments. Thank you very much. Coming to question number 1, a scale bar on an electron micrograph is 2 centimeter long. That's the important thing. So 2 centimeter is equal to 20 millimeter, and then 20 millimeter is equal to 20,000 micrometer. But then this 2 centimeter was equal to 1 micrometer. So you can understand how the magnification would be this. So 2 centimeter converted to the unit which is given to you. If it was a nanometer, then you would have said what? You would have said, okay, 2 centimeter is equal to 20 millimeter, which is equal to 20,000 micrometer, and then another 3 zeros for nanometer. So then this would have been the magnification if this had been nanometers instead of micrometer. So an easy way of doing this is convert it to the same unit and then decide what was the magnification. And then coming to question number 2, the eyepiece of a microscope is fitted with eyepiece graticule and a stage micrometer scale is placed on the microscope. Well, that is, you only place it in order to calibrate it. And then you remove the stage micrometer, and then you can put a blood slide or a leaf slide or a stem slide and then measure the actual cells. So which statement for the stage micrometer scale are correct? Which statements are correct? The scale can be used to measure the actual length of cells directly. Well, you don't do it directly. You have to remove the stage micrometer and then place another slide, because you have to first calibrate the microscope. The scale allows you to calibrate the eyepiece graticule. Yes, that's correct. Less of the scale is visible as the objective lens changes from x10 to x40. Naturally, if you change the objective lens, you will see less of the scale, because now less of the view is there. So that is why the answer to question number 2 was B, which is 2 and 3 only. This was incorrect because you don't actually see it directly, but you actually have to remove it and place the slide on it. Then coming to question number 3, 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. mRNA passes through to the ribosome. So this would be the nuclear membrane. So the mRNA, which is formed in the nucleus and passes through the nuclear pores, then organize microtubules to produce the spindle. Now this is of course the centrioles. Organize microtubules to produce spindle during cell division. Then synthesis of polypeptides would be on the ribosomes, because mRNA goes and attaches on the ribosome, and then of course the tRNAs bring the appropriate amino acids and a polypeptide chain will be constructed. The appearances were listed by letter. V, membrane which surrounds an enclosed inner cavity. V, membranes which surround an enclosed inner cavity. Non-membrane bound spherical structures. Now that would be the ribosome, because ribosomes are not membrane bound organelles. A double membrane interspersed with pores. Double membrane has to be the nuclear membrane, the nuclear envelope or the nuclear membrane. It's a double membrane. Non-membrane bound cylindrical structures would be the centrioles. Membrane bound sacs arranged as flattened stack. That's the Golgi body. That's how we describe them. That's the English that we use. So which student correctly matched the numbered function with the appearance of the cell structure? Now the first one was mRNA passes through the ribosomes. So 1 should have been X. So then we realized that these two could have been correct. Then what was 2? 2 was organized microtubules. Now that would be the non-membrane bound cylindrical structures. This one. And then number 3 was synthesis of polypeptides. So that is on the ribosome. Polypeptides are made on the ribosome. So the ribosomes were what? The ribosomes were this non-membrane bound spherical structures. Ribosomes have, they're found on the RER. So they are non-membrane bound structures. Membrane bound are mitochondria, chloroplasts, smooth ER, RER, Golgi body. So this is actually a part of the syllabus which they expect you to know which one is membrane bound and which one is non-membrane bound. So that is why the answer to this was D, which is the one part mRNA passes through nuclear membrane, then Y was the centriole, and then W was the ribosome. Coming to another difficult question, which cell structures contain ribosomal RNA? You see, ribosomal RNA would be in ribosomes. Ribosomes are made up of ribosomal RNA. And where is the ribosomal RNA made? It's made in the nucleolus from the DNA. You see, the latest definition of gene is a portion of DNA which codes for a protein or a nucleic acid. Because where do the tRNAs come? Where do the rRNAs come? They also are coded in the DNA. So which structures contain RNA? You have to understand wherever there is ribosomes. So chloroplasts have ribosomes, 70S, mitochondria have ribosomes, but this does not have any ribosomes. So that is why the answer to this was B. Nucleus also has, in the nucleolus, the ribosomes are made. In the nucleus, the ribosomes are made, so there will be ribosomal RNA in the nucleus as well. So the answer to this was 1, 2, and 3. Question. A student carried out 4 tests on a sample of biological molecules. Now, if it was cloudy, this means fat was present. If Benedict's test showed yellow, that means reducing sugar is present. That means reducing sugar is present, not glucose, fructose. If this is purple, that means protein is present, not that enzymes are present. Because proteins can be enzymes, can be hemoglobin, can be myoglobin, can be antibodies, can be anything. But if the biuret test is purple, it means proteins are present. Doesn't mean amylase or pepsin is present. It just means a protein is present. Iodine is yellow, means no starch is present. This was the catch here was this. You all thought, as it says in the syllabus, that it says in the exam report, that a large number of students assumed that was glucose, and that was not right. And that is why many of the students got this wrong. Because the simple reason is that when you do tests, you only get to know what is the biological molecule present. You don't get to know the specific biological molecule. So emulsion cloudy means fat present. Benedict's yellow means reducing sugar present. Purple, biuret purple means protein present. Iodine means yellow means no starch present. So that is why what we have to understand is when we are reading the question, we've got to be very careful. So the answer was 1, 3, and 4. Fat was present, protein was present, starch was not present. But it said reducing sugar was present. We didn't know it was glucose or fructose or whatever. So this was the catch in it, that you were not reading this question carefully. And you were just looking at it and saying, okay, well, of course, if it's Benedict's is yellow, that means glucose is present. Why? It means reducing sugar is present, not glucose is present. Which molecules contain at least 2 double bonds? Saturated fatty acid, collagen, hemoglobin, collagen, saturated fatty acid, hemoglobin and collagen, saturated fatty acid. Now the question was, what are saturated fatty acids and what are unsaturated fatty acids? So you see here 3, we've got saturated fatty acids in 3. That was a clue for you. All those were wrong. But why is the answer C? Which is, you've got to understand, why is the answer C, hemoglobin and collagen only? Because saturated fatty acids do not have a double bond. Now here I'm showing you this diagram of a saturated and an unsaturated fatty acid. So where is a double bond in a saturated fatty acid? And of course you remember in a phospholipid molecule, when we're drawing the phospholipid molecule, we draw it like this. Why do we draw these crooked? It's because it has a double bond here, and the double bond makes it to have a kink in it. If I've drawn this like this straight, then that means it's a saturated fatty acid present. So unsaturated fatty acids would have a double bond. Saturated fatty acids do not have a double bond. Now coming to question number 13, which row is correct about the structure of proteins? Now whenever you get such a table, you've got to figure out where there is a mistake in it. And this is a difficult question. So which row is correct about the structure of proteins? A is primarily determined by the sequence of DNA nucleotides. Occurs by hydrogen bonding between NH and CO, disulfide ionic hydrogen. This is correct. This is correct. This is correct. Always formed by association of two or more polypeptides. Yes, that is correct. So now the important thing is that it is not that which one was correct. I know the answer is A, but why is B wrong? Now that is what you have got to decide in this. Now it says in the exam report, less than half of all candidates answered this correctly and almost a third incorrectly, selecting option D. Now why is D wrong? Why is D wrong is because here this is wrong. Always formed by two or more polypeptides and a metal ion. No, a metal ion is not present in not all protein molecules have a metal ion in the quaternary structure. So you've got to pick up the ones which are wrong. Now, for instance, in the C one, this was wrong. Only globular proteins have their level of structure. No, they can be present in any other proteins as well. Fibrous proteins also have tertiary structures. Then in B, maybe some of you might not agree with me, but then the thing is, I would have thought this one was not very correct because the number of amino acids in each polypeptide chain is not the number, it's the sequence of amino acids, which is the definition of the primary structure. I always say number and sequence, but basically it is the sequence which is important in the primary structure. So if B, C, and D were wrong, why were they wrong? This is what you must understand is whenever you're doing these questions is to figure out which ones are wrong and then sort of mark those and then say come on to narrow down to the correct choice. But this was a difficult question. The graph shows the relationship between the concentration of substance and the rate of an enzyme catalyzed reaction. Which row shows Km and Vmax for this enzyme would be effective if the same reaction was carried out in the presence of a competitive inhibitor. In a competitive inhibitor, it will finally reach the Vmax. So the Vmax would remain the same. But what would happen to the Km? So we have to take half. So this was the Km initially. Now what would be the Km? The Km would be now here. So the Km would have increased. So Km would have increased. So that is why what we have to understand is the answer would be B. But you have to understand why, because you see when a competitive inhibitor is added, finally it does reach the Vmax. So Vmax would remain the same. But what would happen to the Km? This is what you've got to figure out. Now, coming to next question, which is a difficult question, 16. Liver cells contain vesicles that are proteins in their membranes, which are specific for the transport of glucose. When these cells need to take up glucose, the vesicles fuse with the cell membrane. Vesicle means this. A vesicle means it has a cell membrane and then it will come and fuse with the cell surface membrane. How does the uptake of glucose occur? Well, glucose cannot enter. It's a polar. It's water soluble, so it cannot enter. It has to enter by facilitated diffusion. It has to enter through a channel protein. So you can see here, these vesicles, if you have done A2, you would know this very well. We have these GLUT proteins. So when these cells need to take up glucose, the vesicles fuse with the cell membrane. The vesicle is this vesicle, which is the cell membrane, but it contains these channel proteins for glucose. And this is going to fuse with the cell membrane, and that is going to result in facilitated diffusion. You have to understand it was basically asking you the form of movement of molecules, which is chapter 4 of the cell membrane. A very difficult question. Candidates found this very difficult and all the agar blocks are stated uniformly. Therefore, the original block of 1 into 1 into 1. So, this was a difficult question. Let's read through it now. A student set up an experiment to investigate diffusion. A block of agar 1 into 1 into 1. So, that means the volume is 1 cm cube. First, let's get this right. Was stained uniformly with a water-soluble blue dye. The block of agar was put into a test tube containing 10 cm distilled water at 20 degrees Celsius. The intensity of the blue color. So some sort of a blue color diffused out. The intensity of the blue color of the water after 5 minutes was measured. Okay, so you have a test tube in which you have put a cube in it, which is colored with a blue dye, and you wait for the blue dye to diffuse out, and then this intensity of the blue color after 5 minutes was measured. So how dark or how light was this blue color? 4 other experiments A, B, C, D were carried out using different numbers of agar blocks, different sizes of agar blocks, different numbers and different sizes. You can see the numbers are different and the sizes are different as well. And different temperatures. All other variables were standardized. Which experiment would give a lighter blue color? So this blue color, this dark blue color or a light blue color? The dark blue color or a light blue color in the water after 5 minutes compared to the first experiment. So now let's look at the calculations. So A is zero point 0.5 into 0.5 into 0.5, so it will be 0.125. B is 0.5 into 0.5 is be 0.125. But then there are 2 of these and there are 8 of these. So if you look at then C, it is 0.5 and then D is 2. So this is the total volume, but then you have to multiply it by the number of agar blocks. Now in A and C, what do we see? In A and C, it is 0.25 into 8, so it's 1. Now the volume is the same. What we've got to understand is that the volume in A and C is the same. Now what we have to understand is in the options at 30 degrees Celsius, the volume of uniformly stained agar is the same as the original block. Therefore, the dye in the water will be at least the same color as in the original experiment, if not darker blue. But we were asked which experiment would give a lighter blue color. So A and C are wrong. Now we are left with D. D ka to, the volume is too much, 2 cm cube. The volume has to be the same as 1 cm cube. So that is why you're left with, in question number 17, you're left with the option B, which was the correct answer. But please understand it, why was this the correct answer? Because you see, if you look at this, it is the same volume, but of course the 2 agar blocks, and of course then what would happen is that there's less. At 20 degrees Celsius in option B, there is only a quarter of the volume. Yes, it's only 0.125. It's not the same. Sorry, I missed that out in the beginning. I said something wrong. So the volume is less, and what we have to understand, it's 0.125 as compared to 1 cm cube. So at 20 degrees in option B, there's only a quarter of the volume of the uniformly stained agar. So as this diffuses out, the water will be a lighter blue in color. The water will be a lighter blue in color because of the fact that the volume of it is less than the original agar block volume. So 0.125 into 2 would be 0.5 because we've used 2 agar blocks. So 0.5 into 0.5 into 0.5, 0.125 and then 2, so 0.5, but the original was 1 cm cube. So this is how you would have to do this question. This was technically a little difficult question. I've tried to make it easier for you. I hope this helps you all. If this doesn't help you, please look at the exam report for this paper. Then coming to another difficult question, the photomicrograph shows cells in various stages of the cell cycle. So this would be metaphase, this would be telophase, sort of the nucleus is coming to form. This would be anaphase, and this would be prophase. So which cells contain, it says, shows various stages of the cell cycle. Which cell contains twice as many DNA molecules as a cell from the same organism that has just finished a complete mitotic cell cycle ending with cytokinesis? So if we had original 10 chromosomes, then you know in telophase what is going to happen. There are going to be 2 nucleuses with 10 in each. So one cell has 10 in each, and then 2 cells are going to be formed with 10 in each. But during this part, what has happened? In metaphase also they are double. In telophase also they are double, in prophase they are also double, in anaphase they are also double. So the answer to this was A, but I'm going to now clarify this point to you all because I can expect a very difficult MCQ on this. So when are they going to be double and when are they going to be single? So I'm just going to share a diagram with you. Now, if you look at this diagram of the cell cycle, what you've got to understand in G1, what is happening? Cellular contents excluding the chromosomes are duplicated, so organelles replicate. Then here is the doubling starts. So if you had, say, human beings have 46 chromosomes, so now they are 92 chromatids. Now they remain 92. Now similarly here in prophase, they remain 92. In metaphase, they remain 92. In anaphase, they remain 92. Then in telophase, they remain 92. So they will remain 92 in all these four different stages: prophase, metaphase, and anaphase. And this, of course, is the mitosis, which is the nuclear division. And then now this is not part of nuclear division. When cytokinesis takes place, that is when the cell divides into two. And now they will have 46 in each. So basically you have 92 in all throughout. In S phase, G2, 92 prophase, metaphase, anaphase, and telophase. And then when cytokinesis occurs, then it will go to half the original chromosome number will be restored. Then a slightly difficult question: uncontrolled cell division can result in the formation of a tumor. Which part of the cell cycle would take less time during the formation of a tumor? You see, cells divide by mitosis. So if a cell divides by mitosis, then what is going to be the prophase, metaphase, anaphase, and telophase are going to be much more faster. But then the other phase, which is of course this phase, the interphase, is going to take lesser time. The grow G1, S, and G2 is going to take less time because we're going to have uncontrolled cell division. That means more mitosis, more cytokinesis. But then the interphase would take less time because we are saying that this is going to be more rapid, and that is what results in a tumor. Another very interesting question. A piece of DNA molecules contain 84 base pairs. The table shows the number of adenines and cytosine bases in one or both of the DNA strands. So first of all, let's add these: 28 plus 23 plus 15. Now how much is this? 66. So you minus that, 84 minus 66, and that is 18. So here we have 18 here. Now it says, but it's not asking you this, it says how many guanine bases are present in this piece of DNA molecule. How many guanine means, so how many cytosine do we have? We have cytosine 18 and 15. So the answer to this was 33. But why is this 33? This is a little confusing for you all, and I want to explain this to you all. If you look at this, this is the way I would have drawn this. Strand 1 is A is 28, C is 15, and T is 23. So then if C is 15, then guanine is 15 here. And if A is 28, then T is 28 here. But then we were left with the 18. So 18 was the one which was the next thing. So 18 plus 15, that would give you the 33. Now, question number 23 was a little difficult as well. The mRNA sequences of the 3 stop codons are shown. So immediately what I do is when mRNA is given, then I will calculate the DNA or the tRNA, whatever the question says. So if you look at the DNA from mRNA UAA, the DNA is ATT. Then UAG, the DNA is ATC. And for UGA, it is ACT. Now, which mutation in the template strand, transcribed strand of a DNA sequence that codes for a polypeptide would cause translation to stop prematurely? So whichever one becomes, so you've got ATT, ATCA. So here is an ATC and here is an ATT. Now, why is the answer A wrong? Because if ATT was present in the template strand already, then it would have resulted in a stop codon. So if ATT was present, it said which mutation in the template strand that code would cause translation to stop. If it was ATT, it would have resulted in a stop codon even then. But it said mutation has resulted in something changing to something and resulting in the translation being stopped. So that is why the answer was C. A was wrong for this reason, but many of you, of course, gave A, but you didn't realize that if ATT was present in the original transcribed strand, then of course they would have not been, that would have stopped the translation then. So the mutation would not have been, it was just present there before. It wasn't a mutation in the template strand. So this was a slight catch, and everybody should be careful when reading the question. Now another difficult question, which it says in the exam report, it says less than half of all candidates were able to work out the correct answer. So that's a difficult question. What explains how the maximum volume of oxygen is taken up as blood passes through the capillaries of the lungs? Each hemoglobin molecule temporarily binds to four, no, it binds to 8 oxygen atoms. The carboxyhemoglobin story has to be wrong because carboxyhemoglobin is only formed when carbon monoxide. Then the binding of the first oxygen molecule to hemoglobin decreases the affinity. No, it increases the affinity. You see, it is an allosteric effect which increases the affinity, and more of the oxygen binds quickly to it. So this was wrong if you didn't know your subject matter. And of course, then that is mean the only answer possible was D. So, but we have to understand why these three were wrong. These three were wrong. Here it's 8 atoms, here it's carboxy, which is wrong, and then of course the binding decreases, it increases. Last question, which row shows the tissues that are present in the wall of the trachea and the wall of the bronchus? Now you've got to understand squamous epithelium. Where is squamous epithelium present? We have to be clear on this. Goblet cells and smooth muscles, of course, are present in it. So the answer was C, but then what are squamous epithelium? I just want to show you a diagram where squamous epithelium is present. Now, the different epithelial cells are squamous, cuboidal, columnar, and ciliated columnar. Of course, you don't have to know these names, but just to get you a different idea of these, how do they look like and where are they found? Well, they're found in the alveoli and they're found in the lining of the blood vessels. And that is the important thing. You've got to understand how they look like and how they're a flattened layer of cells. And the squamous epithelium is not present in the trachea or the bronchi. It's ciliated epithelium, which is present in the trachea and the bronchi. Thank you for watching these videos, and thank you for subscribing, and wish you all the very best.